I don't know what day of the week it is. Okay. So quickly, to finish up chapter 16, I need to introduce to you a couple more molecular machines. So on Thursday, we went through the bulk of chapter 16, and we talked about the 4 steps of DNA replication. What is step 1?
Oh, that's good.
Step 1 of DNA replication. Somebody remind me.
Unzip
the 2 strands. Unzip the 2 strands. And what molecular machine unzips the 2 strands? Helicase. Love it. Okay. What is step 2? Rick.
Copy short stretch as RNA.
Step 2 is to copy a short stretch of the template as RNA. And what molecular machine does step 2? Katie.
Primase.
Primase. Step 3. What is step 3? Michelle. Okay, so allow something to take over and start copying it as DNA nucleotides. Okay, and what molecular machine does step 3? Emma? DNA polymerase. And specifically DNA polymerase 3. And then step 4 is what?
Replace RNA.
Yes, but replace the RNA nucleotides and then fix all the nicks. And that step is done by 2 molecular machines. DNA ligase to fix the nicks. DNA polymerase 1 to cut out all the RNA nucleotides. Kyle.
What was the differences between all the polymerases?
Well, DNA polymerase 1 and 3 both write in the same direction, and that is 5' to 3'. And they can't add the first nucleotide. The difference is DNA polymerase 1 can cut out the RNA nucleotides that were written by primase. DNA polymerase 3 cannot. Okay? DNA polymerase 3 cannot. Okay. I need to give you 2 more molecular machines. Okay? First molecular machine. Single-stranded.
Did you forget an E?
Yes, I did. Single-stranded binding protein. Yes, I did forget an E. Thank you all for always pointing out my mistakes. Yeah, Mark.
Do you have record on?
I do.
Okay.
You bet I do.
Just making sure it's just
Hold on, what is this?
It's one of the 2 remaining molecular machines that we need to talk about.
Okay.
So single-stranded binding proteins bind the strands. I know. After helicase unzips them to prevent them reconnecting before steps 2 through 4. And what are steps 2 through 4 again? Write a short RNA section, write DNA nucleotides, replace the RNA, fix all the nicks. Okay? So, after step 1, when helicase unzips those 2 strands, there's a time before steps 2 through 4 happen that these single-stranded binding proteins bind to those strands and keep them from reconnecting before steps 2 through 4 happen. Alright? Does that make sense? Alright. Cool. The last molecular machine. DNA gyrase / topoisomerase. 2 names for the same molecular machine. DNA gyrase / topoisomerase. You like that name?
This sounds like a different language.
It is. It's molecular biology.
It's the language of science.
I know, but that's why.
Oh my gosh,
That is from Madagascar. Are you serious? Wow.
You stink.
Wow. Okay.
Yeah.
Is that a Russell Martin person?
Relax, it is. Oh yeah. It's old.
So
But he's back.
I know.
Okay. Okay.
It's better than just.
DNA gyrase topoisomerase. So I want you to think about this for a minute. You've got a braided stretch of hair. Okay? You've got hair that's in a braid, either on your own head or someone else's, and you start to pull the braid apart up right where the braid connects with the head. Okay? So you've got a braid, right? And you're starting to pull the braid apart right where it connects with the head. What happens? Yeah, it gets tangled. You create enormous torsional strain down below where you're trying to pull it apart, right?
Yeah.
Because you've got to pull it apart at the end and work your way up. Okay, when you start to pull the two strands of DNA apart, it creates enormous torsional strain on either side of wherever that replication bubble is. Okay, when you start to pull those two strands apart, it overwinds everything on either side of that DNA on that replication bubble. Does that make sense? You're starting to pull those strands apart just like trying to undo a braid by pulling it out from the top of the head. It's fun to do, as long as it's not your braid, but it doesn't work very well. Okay, so what DNA gyrase does, hold off of your, I know you're not going to forget your story. Okay.
I got a story.
So DNA gyrase topoisomerase eliminates supercoiling on either side of the bubble. On either side of the replication bubble. And do you know what I mean when I say replication bubble? So the place at which helicase is currently unzipping them, you have basically normal DNA on either side of that, and as you start pulling those two strands apart, it supercoils on either side of that bubble. And so DNA gyrase / topoisomerase eliminates that supercoiling. Okay. Isa.
Okay. So I have GB. So it's a wild thing. So these two girls got into a fight. These two black girls, and that's for it.
Pour it again.
Wait.
Tori.
Can I see my grade?
We'll do it at the end. Okay. All right. Chapter 17. Any questions on chapter 16?
Chapter 17?
Yep. Chapter 16 is done now. You all have seen how replication works.
Alright, so
Chapter 17, which is called gene expression.
Thanks, Doctor.
You bet. Chapter 17. Gene expression. I don't feel like you all are trying very hard not to talk in this latter half of the course.
Gene expression.
This is what chapter 7 is called. Gene expression, gene to protein.
What's your favorite punctuation?
That's
exactly what he doesn't want us to do.
I know.
I don't have one.
Ow, my head.
Okay.
Alright, we're ready.
Okay. Chapter 17, gene to protein. And so in chapter 17, we have what's called the central dogma
The biology.
of molecular biology.
DNA RNA.
The central dogma of molecular biology. And here's what the central dogma says. You have one gene, and that is found in the DNA. And that one gene is transcribed, transcription, into one RNA, which is then translated. Wow, you are not very good at this. My kids would destroy you in the quiet game.
Oh, I'm bombing quiet. I'm good.
Okay.
Yeah.
So we've got one gene found in the DNA is transcribed into one RNA, which is then translated into one protein. This is the central dogma of molecular biology. You basically have one gene, one code in the DNA, and it gets transcribed or written into one version of RNA, which is then translated into one protein. What does it mean to transcribe?
To rewrite.
It means to write, right? And so here the language is not changing. We are writing from a nucleic acid into a nucleic acid. Now the letters are a little bit different. It's akin to, instead of writing in black here, we're going to write in blue. But the language is the same. Okay? But to translate, this is a very different meaning. Now the language is changing. Okay? This is the central dogma of molecular biology. One gene transcribed into one RNA. It's translated into one protein. By the time we did the Human Genome Project, which published all of its findings in 2002 when they sequenced the human genome.
Oh, recent.
Yep. Sort of. Almost 20 years. 2002, that was like the year some of you were born. Some of you were born in 2003. So the year of your birth marked... Nobody here...
Who was born?
Oh, so some of them are 2001. Okay. Okay. And so by this time, we knew that there were approximately 2 million proteins functioning in human cells. About 2 million. So according to the central dogma of molecular biology, if there are 2 million proteins functioning in human cells, how many genes should there be?
2 million.
2 million. Do you know how many we found when we did the Human Genome Project? Somebody take a guess.
17 million.
Okay, so we've got more than 2 million. We've got, you said 17 million?
I say 2 million.
Okay, so you're going to go 2 million. You're going to go 17 million. You said more than 2 million. Pick another.
So much more.
So much more, but not so much more.
I think a lot.
So how about 5?
I think there's more protein.
About 10 million.
Is that okay?
Are we
Okay.
saying, what are we predicting?
So, Carrie goes, Carrie says 5 million.
Wait, oh, okay.
You right?
Sure, 3 million.
Carrie says 3 million.
I thought you
What
meant
number
like 2
are we
million with like
predicting?
5. Like
Katie.
2 million and 5. I was like,
Katie says 2 million. Isa says 17 million. Lydia says 1 million. Do we have any other votes?
What are we
7 million. 1 million.
The number of genes. If we've got 2 million, we have 2 million proteins functioning in human cells. According to the central dogma of molecular biology, we should have 2 million genes.
Yeah.
Okay, so we've got a vote for 2 million. We have a vote for 1 million. We have a vote for 17 million. A vote for 2.9 million. Okay, Kyle. 6 million. Mark.
6 billion.
2 billion.
6.
10 million. Okay, look at this. I love this. Your all predictions are somewhat in line with what people thought they would find when they did the Human Genome Project.
6. I swear it's 6.
21,000.
I was gonna say less, but then I decided not to.
21,000.
Oh, is that because we like protein in our diet and stuff?
That's 100 times fewer genes than what you would predict according to the central dogma of molecular biology.
But then
What this means is, go ahead.
how does it still work?
I'll show you. Okay, I'll show you. So what this means, no, you don't get them from other places. You get all of your proteins from these 21,000 genes. So what this means is the average gene codes for 100 different proteins. The average gene, there's about 21,000 of them in the human genome, codes for 100 different proteins. The average gene. We're like, this doesn't make any sense according to the central dogma of molecular biology. Which means that the central dogma of molecular biology is not right.
Whoa.
Is not right. Okay, let me show you this too.
They lied to us. The audacity.
We were talking about this.
You were talking.
Okay, so from these 21,000 genes, we generate 2 million different proteins. Michelle.
What is Dogma means?
Dogma is like truth. Yeah, like the core truths. The core beliefs.
It's a long
The movie Dogma? Is that what you're talking about?
I'm talking about the I seen green.
Oh.
But as a
Like a
dog.
dog's
Okay.
mouth.
Is it about dogs
So these 21,000 genes take up about 1.2% of the human genome. 1.2% of the human genome is made up of these 21,000 genes. 1.2%. Nobody expected this. Following nobody expected to find this when we did that when we started the Human Genome Project. So according to the Central Dogma of Molecular Biology, 98.8% of the human genome is junk. Doesn't do anything.
Then why is it there?
Right? Well, that's an excellent question. So starting in about 2005, people are like, there's no way this can be junk. We need to figure out what this 98.8% does.
Yeah.
Okay? So we already know that the central dogma is not right because it's not one gene makes one RNA makes one protein. It's actually one gene makes 100 different RNAs that make 100 different proteins. Okay?
With 1,000 proteins.
Why'd you test the wrong one?
What's that? Why did I test the wrong? What?
Why you told us the wrong one?
Well, because I gave you the central dogma of molecular biology to show you that it's not right.
I mean, we should be able to assume that the
Okay.
central dogma of something is true, but you know, it's fine.
Okay, so 1.2% of the genome is made up of these 21,000 genes. Another reason the central dogma of molecular biology is wrong is 75% of the genome goes through this step here, but only 1.2% of the genome does this step here.
Where does it go?
So 75% of the genome is transcribed into RNA.
What about the other 25?
That's an excellent question.
Wait, so are you saying 1.2% of the 75?
No, 1.2% of the total. This is 75% of the total goes through this. 1.2% of the total goes through that step. So not only is the central dogma of molecular biology wrong because it's not one gene to one RNA to one protein, it's more like one gene to 100 RNAs to 100 proteins. Also, the central dogma of molecular biology is wrong because 3/4 of the DNA is transcribed, but only 1.2% of it is ever translated. And what does it do? We'll see in just a moment what some of these other RNAs do. What the rest of it does, if you want to know what does the other 25% do, Issa, did you ask that? The other 25%, some of it is telomeres at the ends of chromosomes, allowing those chromosomes to shorten without losing information.
Telomerase.
Right? Well, telomerase is a different beast altogether.
Beast.
Okay? So, but those telomeres, they shorten every time you replicate. So the cells that you're making in your body now, they're starting off with shorter chromosomes than the cells you made 16 years ago. Okay? But they're still okay because the ends of your chromosomes are called telomeres and they just have the same section repeated over and over and over and over again so it can shorten and you're not losing any information. So some of them are that. You also know on chromosomes we have the places where the two sister chromatids attach together. What do we call those? Well, we call them sister chromatids, but in which region on those chromosomes do they attach together? Kyle, where do the sister chromatids attach? The centromere. So part of this 25% is also the centromeres, the middle of the chromosomes that have to match up perfectly so that chromosome 1 and chromosome 1 from your mom and chromosome 1 from your dad can interact together. Emma.
When was this dogma established?
In the 1930s, 1940s. You're like, nothing that happened in the 1930s and 1940s was true.
Of course not.
I mean, it was like jazz, awful.
I love jazz.
I do too.
love
I love
Do you
it.
like Billie Holiday?
Sometimes.
Oh my gosh.
So, what else happened in the 1930s and 1940s? Well, during the 1940s, we had World War II, and so you had women playing professional baseball. That was pretty cool.
A league of their own.
A league of their own? Yeah,
You
Emma.
know, Babe Ruth's daughter, her last living daughter died yesterday.
Really?
At 102 years old.
At 102 years old. You know, Babe Ruth, listen to this. Listen to this, everyone. This has nothing to do with AP Biology except for that Babe Ruth was a living being. Babe Ruth was the first professional athlete to make more than the President of the United States. Now the President of the United States makes less than the league minimum in Major League Baseball. About 400 and something thousand, 430, 440, 450,000.
Yeah.
It's a hard job, let me tell you. I mean,
Let me tell you.
No, but you can. I'm not old enough yet to run for president. You have to be 35.
Hey, would you do it?
Huh?
Would you do it?
31.
If you could.
I know, surprise, right? You're like,
Wait, so MLB players are making more money than the president?
what's that?
MLB players make more.
Oh yeah, people making like. If you're on like a big league salary, like you're on a big league roster, everybody on a big league roster makes more than the president of the United States. Okay. Shh. Did you have a question, Emma or Trinity? Somebody had a hand up over here. Okay. Okay. So listen, the central dogma of molecular biology in no way can explain what's going on inside of human cells. First of all, it's way more complicated than one gene is one RNA is one protein. Second of all, we've got a lot of RNAs that are never translated into protein. They get transcribed into RNA and they do something else. They don't get translated into protein. They do something else. Yeah. What do? They regulate this process. They regulate the making of proteins. Because once you make a protein, that protein's going to start to do its work. Right? The proteins, these are the, the components that are doing the work of the cell. Oh man! So once you make that protein, it's over unless you mark the proteins for destruction. And so you have to control how you make those proteins, when you make those proteins, how much of those proteins you make. It's like once you make children, it's over, right? It's over. Once you birth those children, your life is different. It's not over. It's just different, okay? All right. Does this make sense? Shh. Any questions here? Any questions here?
What's correct?
That is the day where I was no longer a child.
I'll just say that the central dogma is it's way too simple. What's written right here is what's correct. That one gene can actually code for 100 different RNAs that can be translated into 100 different proteins. Okay? And then, you know, a lot of the DNA is transcribed into RNA and never gets translated. Okay? All right. Any questions, thoughts, concerns? We've got a few minutes. We're going to go for about another 8 or 9 minutes or so, and then we'll stop. And if you want to actually see your score on the exam, that you didn't look it up at RedWeb, I can show you that.
I'm going to be the one who got the 2.
All right.
Starting from the top, now we hit the bottom.
Wait, so only 2 people got 4s, right?
3.
3 people.
3 people got 4s. 2 people were right on the bridge between a 4 and a 5. Okay. All right. No questions? All right. You know what? 5 people.
You see?
5 people got 4s?
Oh.
You didn't say anything yet.
Dude, what if one of them was me?
I don't know. I don't know. Now I don't know.
Wait, what's the equivalent? I definitely failed that. Like an 85?
Yeah.
Oh, points? Oh, you mean percentage? Oh, so what I did this time is if you got a 5, I gave you 100% on the exam.
Wait,
but nobody did.
Now you do that. Now you do that.
But and then a 4, you got a 90%. A 3, you got an 80%. A 2, you got a 70%. And nobody scored below a 2. But then,
I can live with a 90.
so for some of you, you got a 3, but you got a 3, and your multiple choice was in position to be a 4. So if that happened, because a 3 would be 80%, right? So 80% of 35 is 28. But if you scored 33 on the multiple choice, I gave you 33 on that section and a 28 on the essay. So then you ended up with like, you
So
know.
like a 3 is in place
Like
of
a
a 4 in this
3.5.
case scenario, and a 4 is in the middle.
Yeah.
Now he starts.
All right.
It's fine. It is fine.
Okay.
I really
So we need to talk about these processes in more detail. Transcription. Translation. Yeah, so nobody scored below a 70% on the exam.
Oh, really? Okay, that's
Yeah.
Rico mode.
And most of you didn't score below an 80% because you got what would have been at least a 3. Okay, so transcription.
So technically everyone passed.
Yes. So in transcription, we go from DNA to RNA. In transcription, we go from DNA to RNA, and transcription consists of 3 steps. Three steps of transcription. You ready for them?
Yeah, always.
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 Initiation,
I'm not feeling it.
elongation, and termination. Termination. Initiation, elongation, and termination. Those are our 3 steps of transcription.
How much are we gonna write?
We'll write a little bit under each. Didn't you notice I left space?
Like, what? So there's space to write stuff under
You
there?
don't have to write it underneath. You don't have to copy.
Fine. We'll come back to this later on. Okay. Translation. RNA to protein.
Elongation.
And it is 3 steps. 3 steps to go from RNA to protein in this translation. You ready?
Always.
Initiation,
It's gonna be the same thing.
elongation,
Except the last one's gonna be different.
termination.
I can't vouch for that.
Look at that. Look at that.
It's the same.
It's the same. Transcription, DNA to RNA. Initiation, elongation, termination. Okay, now let me tell you this. Transcription is very similar to DNA replication, except you're only replicating one of the 2 strands. In transcription, it's very similar to replicating the DNA, except you have 2 main differences. One, you're only replicating one of the 2 strands, not both, and you're writing it entirely as RNA. No, you're not doing a short stretch as RNA and then allowing DNA nucleotides to take over. It's all RNA. Other than that, it's very similar. You do the same thing. If you take your chromosome, here, let me take it. Okay, so you take your chromosome and you're like, this portion of this chromosome needs to be transcribed so we can translate a protein from it. So what you do is you just write all in blue the complementary portion to that, and then that's your RNA that can leave and be translated. It's beautiful. Beautiful. Okay, transcription. This happens inside the nucleus. Translation, this happens in the cytosol, in that fluid of the cell on ribosomes.
Yeah, because of
So one's in
But
the
y'all remember this.
the other one's in the what?
Sir.
So this transcription happens within the nucleus. Translation happens in the cytosol on ribosomes.
That's why they're like all over the place, right? So
Yeah, or those ribosomes could be attached to the endoplasmic reticulum. Okay, so transcription, very similar to copying the DNA. Now, this is all fine and good. Now, the central dogma of molecular biology would tell us that you go here to here, and then automatically you go here to here. We know that's not true. Because 75% of the DNA does go from here to here, but most of it never does this. It only does the transcription, never the translation. Only the transcription, never the translation, right? Right? Yeah. So we already know that, but also we know that it's not one gene to one RNA to one protein, right? It's more like one gene to 100 RNAs to 100 different proteins. Meaning we can't just go boom, boom, and then go immediately boom, boom. There has to be something in the middle to where we can take this and modify it and make different RNAs, right? Were you all able to decipher that? I know sometimes I'm hard to understand. My wife asked me a question yesterday and I answered it and she's like, I have no idea what she said. It's so rude of you to answer it like this. My wife? She's a part-time bookkeeper and a homemaker. A stay-at-home mom.
Bookkeeper. I think it should
Like
be a profession.
Alright.
Library.
Right.
I'm gonna major in it.
Okay, so there's something that happens in between these two in eukaryotes. And it's called, check this out. You don't need to actually write it right in between these two because you probably don't have space. But it's called RNA modification. In between these two, we have a step that's called RNA modification. RNA modification. And it is 3 steps. 3 steps.
Why did I write this?
3 steps. In between transcription and translation, you're talking about this?
You know, the way you did it, you were like...
Yes.
This is 3 in German.
So Dirk Nowitzki does that.
What? Yeah. Because they don't do 3 like this. This is a much better way to do 3 anyways. It's way more comfortable.
Because I mean, they used to go like that.
Okay.
Like when they show them, it does something like that. Dirk Nowitzki goes.
Show them how you count in China. It's so weird. And then they do
That's how I count.
Okay. So RNA modification, 3 steps. And you're like, it's initiation, it's elongation, and it's termination. It's not. It's capping.
Oh, wait.
It's polyadenylation.
Oh, that sounds really cool.
Adenylation. Oh yeah.
2.0.
Yeah, we've got to do a 2.0, you bet.
Okay, this could be next.
But we don't have to, we're not going to go into the level of detail that we did in like cellular respiration and photosynthesis. Yeah.
So does one matches to all the ones? With the
No, they're all separate. They're all separate processes.
Oh.
Yeah, they're all separate processes.
So the RNA modification
You should have
Between transcription and translation.
but just the 3 steps of the
Yes.
Okay.
Okay.
And then match to
No.
Oh, okay.
Okay. Shh. So RNA modification, capping, polyadenylation, these 2 are required for the RNA to leave the nucleus. Okay, these 2 are required for the RNA to leave the nucleus. The third one is how you get different RNA products. And this third one is called alternative splicing. Okay, so steps 1 and 2 are capping and polyadenylation. These are required for your RNA to leave the nucleus. This happens in the nucleus, this happens outside the nucleus. In order for this to leave the nucleus, it has to be capped and it has to be polyadenylated. Okay? The third one increases the variety, alternative splicing. And when you splice wires, you take 2 wires and you attach them together. So alternative splicing, you take 2 pieces of this RNA and you splice them together. And we'll see how this works, but not today, because we're out of time today. But we'll see how this works on Wednesday. So on Wednesday, we're going to go through these processes. We're going to go through 1, 2, and 3. Transcription, RNA modification, and translation in a little bit more detail. Alright? And I hope on Wednesday you really do try to work a little bit harder at not talking the entire time. Are you?